Welcome to Basic Bioinformatics, brought to you by the Sequencing Center.
In this video, we're going to show how to import Illumina paired-end FASTQ files into Geneious Prime. We have quite a few clients who use Geneious for bioinformatics data analysis, and they'll often ask what's the correct way to import paired-end FASTQ files into Geneious. So I'll show you how to do that in this video. So what I have here is we run Windows operating system on Amazon AWS workspaces. It's a virtual OS environment, and as part of that we get WorkDocs, which is a shared file system between your local workstation and the cloud. So I'm going to just open up WorkDocs here, and I've got a couple of files, FASTQ files in gzipped compressed format. And I've renamed them from their standard Illumina naming convention to look like this. One says forward read R1, the other one says reverse read R2. So R1 is always, or typically, the forward read in a paired-end read pair, and the reverse read is often called R2, which is the reverse read in the paired-end read. So these two files go together, the forward and reverse read. Okay, so we're going to try to import both of these into Geneious. And so the first thing we want to do is launch Geneious Prime. We have 2019. I'm pretty sure this is the current version, 2019.1.3, and it takes a second or two to actually load up Geneious Prime. Okay, so once Geneious is loaded up, you get the interface that you see here. I'm not going to go over this. We can talk about the Geneious interface in a different video. I'll just assume you're somewhat familiar with it. And so to import the paired-end files, we go to File and down to Import, and then we choose From File. And if we do that, it'll load up this WorkDocs directory, and there's our forward and reverse read files again. Now we have to choose or pick both of these. On my Mac laptop, I have to shift left mouse click on each file to do that. Your machine may be totally different, how you choose both of them, but it's important to have both of them checked because we're going to import them together. Okay, so then we have them both chosen, and then we just choose Import, and it brings up this dialog box. I'm going to reset this to the defaults because that may be what you see the first time you bring up this import dialog box. And it says FASTQ sequences import. And the first drop-down box above is the read technology, and it's in my case it's Illumina by default. If you actually choose the drop-down box, there's a couple other vendors out there, PacBio and Nanopore long read instruments, and then Sanger 454 solid, and so forth. So we want to choose Illumina for that. And then you have the option of either not pairing these two files or pairing them. We definitely want to use the paired end option. We could load them up individually, but we don't want to do that. We want to have the power of paired-end reads, so we want to choose the paired-end inward pointing option there. If you look at the drop-down box, there's a couple other options, mate pair and so forth, that don't really apply to what we're doing here. So we just go with the paired-end read. In the next drop-down box, the couple options there, the one we want to choose is pairs of files. And it explains what that is. It says, the first sequence in one file is paired with the first sequence in the other file. And what they mean is if you look in the actual FASTQ files, the first record in the forward read file will be paired with the first record in the reverse read file. So the first record and the forward and reverse reads are going to be paired together, and that's what we want. And I believe I have something like 1.6 million reads in the forward read file and 1.6 million reads in the reverse read file. And if you pair those together, you wind up with, I guess, 3.2 million reads. Then finally, the insert size is automatically chosen to be 500 here. I won't explain what insert size is in this video. We'll just leave it at 500. That's roughly the correct number for these paired-end reads. And then we click OK. And then it brings up a progress bar, shows you how it's importing these files into Geneious. Okay, so the file import is done. And if you look along the top bar here, it shows what it actually did. First of all, we can enable or disable the paired-end read import file. It gives it a name, usually it's called paired reads. There's a brief description, paired reads created from the forward and reverse read files. It gives you the size of the file, the absolute path that it used, and so forth. So some metadata there on the actual import. In the second section here, if you click on the Sequence View tab, pretty interesting. It brings up all of the reads that it imported. And if you notice here, the first two are going to be paired. There's these sort of like box whisker plots, one kind of forward-facing, one reverse-facing. So this is actually the first record of the forward read from the forward read file. And the second one is the first record in the reverse read file. So these two are paired together, just like we expect them to be, okay? And then on the right-hand side, it just shows you some more information about the actual read itself. I'm not going to go into any detail here, just to let you know that it looks like it did import these all correctly. You can scroll down to the bottom, and just like I said, there's about 3.2 million reads in there. Paired end, go back to the top, and that's pretty much how you import the paired end reads into Geneious.
Thanks for watching this episode of Basic Bioinformatics. If you found this helpful, hit the like button and we'll see you in the next video.